pleasure to be here. So I'm in the program in applied mathematics at the University of Arizona, but I work with Professor Régis Ferrier, who's an ecologist, and postdoc Antonin Affolter, who is also an ecologist and a planetary scientist. So we develop these global models of Europa. And so what you see right here is the Europa Clipper mission, which is going to launch next year in October, hopefully. And I believe it's the largest, in terms of size, spacecraft that NASA's ever built because of those massive solar panels. Because when we're 5 times away from the sun at Jupiter, I think the brightness scale is like inverse square law, so it's 25 times less bright at that area. So why do we care about Europa? So beneath the ice crust, we have an ocean which potentially contains 2 to 3 times the amount of water as on Earth, which is, when I first discovered that fact, I was just so blown away by that. And we have an energy source from tidal heating, namely, we may have hydrothermal venting as a result of that. We saw that on Enceladus because we saw silicates in the plumes. We haven't confirmed that yet in Europa, but it's very likely based on the amount of tidal energy that we expect. And we also have a rock-ocean interface. So that's very essential for cycling chemicals needed for life. And we have a lot of convection, as Sarah talked about, which is very essential also for those energy gradients. So as a result, we need these models to constrain what we're going to see from the mission. So we had JUICE also, the ESA mission. Let's not forget about that. So we need to model all these different layers. So working off what we did for Enceladus to analyze the Cassini data, we're going to do that here, specifically focusing on the ecosystem aspect. And so what we want to do with that is provide clear constraints on what kind of biosignatures we're going to expect based on a number of different hypotheses of abiotic possibilities, biotic possibilities, and habitable possibilities without life. So as I said, our approach is very multi-level. We are going to first look at the bottom layer, which is the mixing of the core and the ocean, to assess, the chemical structure coming out through the water passing through the core, and what chemicals are going to come through that. We're going to have to assess how that mixes in the water to assess how much chemicals we're going to see coming out of the plumes. And then we also need to take some model organisms from Earth to understand the metabolisms that we might expect, along with the ecological and possible evolutionary dynamics of the life that we're modeling. So with this framework, we're going to estimate the likelihoods of certain data sets that we're going to see via a Bayesian framework. So to intimidate you a little bit, we're going to have some math. So let's talk about the mixing. We look at the dimensionless mixing ratio X and the temperature T. So we have a partial differential equation with spatial and time variables. Solving this partial differential equation gives us this steady state composition and temperature of the mixing layer with the mass flux density. So using that information, we can compute what we're going to see coming out of the plumes by integrating over the entire space. So we integrate, the integrand contains this mass flux density and the concentration of a specific chemical component that we're looking at. So whether that be glycine or other amino acids, or cells potentially. So the concentration of this compound at the base of the ocean plume, assuming that the buoyant mixing layer mixes together as we described from the previous model, is given by this quotient of these integrals. So then we want to look at the cell metabolism and output, and there's a very large variety of possibilities we could be working with here. So we kind of run the model for tons and tons of possibilities of Earth analogs. dependent on these environmental conditions. So we model this catabolic reaction rate of a specific organism using the Arrhenius law. I know that came up yesterday, Arrhenius, in one of the trivia questions. So the Arrhenius law is specifically this quotient here, and we use the catabolic energy constant given by this term, and it goes in here, to understand the ratio of inactivated to activated enzymes for the reaction. Now, critically, we assess the ecological dynamics of the system. So in this case, we are dealing with one population, which is given by a standard ODE model describing the dynamics, but we've been thinking about incorporating multi-species into this because there's actually a significant dynamical difference when you have competition between species in the environment, and that would seriously affect what we'll see when we go there. So we have to have multiple different ecosystem dynamics models. And so we solve this and get a steady state concentration of a specific compound from this. So what can we learn from specifically the Europa Clipper mission? So the Europa Clipper mission has some very interesting instruments. We have a mapping imaging spectrometer. So it'll map the surface and conduct the spectroscopy of the surface and the distribution of the organics. So as things come out of the plumes, they fall onto the surface, and we should expect to see an interesting chemical composition. I know they're already doing this with JWST from a distance. They haven't analyzed that data yet, but should be interesting. And then to sort of sniff the exosphere of Europa, we have a mass spectrometer, mass specs. It'll determine the composition of the ocean beneath by sniffing that exosphere. And then we have a surface dust analyzer. It'll measure like the solid particles coming out. And so in this image, you can see kind of a remote identification of NaCl via spectroscopy. So let's focus on this slide a little bit. So this is the overall framework. So at the top, we have those mechanistic models for the geochemistry and the biology that we discussed earlier. And so the flow of information goes from the top as prior probability densities that flow into as parameters into the general model. Then we pass those parameters into the biological model for physiology and evolution, which then goes to our habitability criterion. So the habitability criterion that we generally use here it's a differential equation describing the population. So essentially, if I were to take an Earth-like organism, drop it onto Europa, would the change in the population be positive or would it be zero? So the derivative of population growth, essentially. And so we pass that into Bayes' theorem as prior distributions. The posterior distributions come from these. Actually, to be more specific, we simulate all of these models with thousands of data points to get what we call pseudo-data in what's called an approximate Bayesian computation. And so we pass that into the model, and we take the observational data, and we put that into Bayes' theorem. So if any of you are aware of Bayes' theorem, that is a way of assessing the probability of a certain event given prior information and given posterior information. And so now we have this cycle of information. So after we get a probability from Bayes' theorem, we can pass it back into the geophysical parameters model and then continue to constrain as we get more observational data. So here's an example of how we did that with Enceladus for Cassini. So in this graph here, you can see the blue dots are simulations for uninhabitable parameter space. The orange dots are uninhabited but habitable. And the green dots are habitable and habited. So what you can see from analyzing the methane content of the plume in Cassini using this framework was that the vast majority of simulations for all the possibilities we assessed fall under the biotic category, which is pretty exciting because, I mean, that speaks for itself, right? So, but there's a very important constraint on this, namely the probability of abiogenesis. So what we do in this model is assess all the possibilities from probability 0 to probability 1 of abiogenesis. And so what we found was at a pretty low probability of abiogenesis, from the previous slide, with a low probability of abiogenesis, you see that biotic explanations are the most probable for what we're seeing in the methane content. So we really need to develop our models of abiogenesis to really limit this variable and constrain this probability. So the conclusion is, in order to find life, we really have to model these planetary systems with the assumption that there could be life there already. Because if we don't, we're going to show up there and we're going to think, okay, it's habitable, but why? Because potentially the habitability of a planet is actually coming from the life that's already there. So we have to incorporate that into our potential models. So this will allow us to really enhance mission data analysis beforehand. So, you know, when we had Cassini, we showed up there and we had no idea what was going on. We're like, wow, plumes, this is amazing. But it took many, many years to really assess this. So hopefully with Europa Clipper, we have this framework in hand as a software package so the data can be fed through real time. And the benefit of this is then we can narrow down in on specific areas of Europa that are of interest to us and really maximize the scientific yield of these missions. So with that, I'll take questions and thank you.
Nice talk. 2 questions maybe. The first, did you choose a prior for abiogenesis?
Yeah, so we sampled the entire space from 0 to 1 probability in those 10,000 or so simulations. So basically, we can't pick a prior for abiogenesis. There's no constraint on that variable. So essentially, we have to run the model for all possibilities of abiogenesis. So like I said, specifically with the Enceladus data, we found that much less than 50% chance abiogenesis ensures that the biotic explanation is the most probable explanation. I believe is around 30% or less probability. But if you're beneath that threshold, then really the abiotic explanations are way more probable, even though the samples from the abiotic simulations are much smaller. But you know, when the probability of abiogenesis is really low, it's just not going to happen, right? So.
Okay, yeah, that answered my second question. Yeah, it's sort of right.
Great.
Hi, thank you for your talk. So for Europa, are you also, in terms of metabolism, are you thinking about methanogenesis or you? Okay.
So that was the first step to consider. For Enceladus, it was kind of the most obvious thing to look at because of the methane content. With Europa, it's more complex because we don't have any real data from Europa. And also the environments on Europa are much more varied. You have subsurface pockets of water along with the ocean, and maybe you have like biofilms on the bottom of the ice. So it's a much more complex thing. So my hope is to run all the simulations for all the possible hypotheses that exist currently.
Okay. And I have one little follow-up question. So you very briefly talked about dynamic communities and you mentioned competition. Are you just looking at competition or are you looking at other sorts of consortiums? Like in a biofilm, there's competition, but there's also lots of sharing going on.
Right. So if we're including multi-species in our models, we're hoping to incorporate all the possible interactions between these species, which is really critical. I want to reiterate because, you know, the competition can kill off like all of the life, you know? So we need to sort of have an evolutionary perspective also, and not just like a real-time ecological perspective to assess these things. So really good question.
Thank you.
Hi, Emily Barris, Stanford University. I was wondering, so in the biological part of the model, what kind of data goes into that, and how can environmental microbiologists help modelers improve their models? What kind of data do you need for that?
Right. So we need Earth analogs primarily. We need the catabolic reaction parameters for specific Earth analogs. So we might want to go to Antarctica and see what's going on there. And that's our best way to constrain it at the moment. So that's how I think biologists really help us in that way. Yeah, exactly.
Thank you so much for your talk, Sam.
Thank you.